probably after the talk, if you look around the room, there's lots of Darwin bowler hats around that are decorated for the day. And that's all of our graduate students who've done such a wonderful job to make today happen. So this is really, we've had several activities today. This is the highlight of our day, which is going to be a wonderful talk by Brian Hall, which, yeah, it is. That's what we were hoping. And so I want to just thank you all for being here to come out and help us celebrate this. And we will have some introductions by Craig Albertson, our local evo-devo guy.
So Betsy said to keep it short, and I went on Brian's. So you know when you have your own Wikipedia site that you got it. So I have some pages here. So I decided that I wasn't going to go an entire overview about Brian's accomplishments, and I was going to be a little bit selfish and talk about the ones that were most influential to me. I'm really pleased to be able to introduce Brian. Dom's read this book apparently, maybe.
That's my cover.
Yeah. With the appropriate acknowledgments, yes. So, so Brian Hall is coming from the University of Dalhousie in Nova Scotia. He has been a giant in the field for as long as I can remember. He got his PhD from the University of New England in New South Wales in 1968. He worked with Patrick Murray, who was himself a very well-known, well-established, famous embryologist. And his PhD research was bone and cartilage differentiation in chick, and that really just launched from there. So it's all been neural crest derivatives and skeletal system, and in many, many influential papers and books. I think one of the first modern incarnations of evo-devo was, and Brian, correct me if I'm wrong, it was your 1975 paper on the evolutionary consequences of skeletal differentiation. And that really is one of the papers that was foundational to bringing evo-devo into the modern era. From there, he wrote a textbook in 1992, Evolutionary Developmental Biology, and then this one, both of which are, again, just standard for anybody getting into this field nowadays. Still standard and still relevant. More recently, Brian has become more generally, he's writing more generally about evolution. He rewrote with Benedict Halgrimsson, Strickberger's Evolution, which I use in my evolution course. Margie Coombs used that for a number of years, and as well as 2010, Evolution Principles and Processes, which is written for a more general scientific audience. And so really, really well written. So awards, and this is where the page, so I'm just going to pick and choose a smattering of awards. So a Fellow of the Royal Society of Canada, 1985, Fry Medal from the Canadian Society of Zoologists, 1994, International Craniofacial Biology Distinguished Science Award, 1996, Canadian Council for the Arts, the Killam Research Fellowship, as well as the Killam Prize in Neural Science. And one of the things that I was struck by, and I think it really speaks to somebody who has influenced the field, is he now within the Canadian Society of Zoologists Division of Comparative Morphology and Development, there is the Hall Prize. So it's a named prize for the best student presentation. So I think that speaks volumes. And so, in other words, to bring it all together, I think we're in for a real treat, and it is my pleasure. And without further ado, I will let Brian tell you a little bit about this.
Thank you, Craig. I'm really glad to be here. I tried really hard to get the zoologist not to name it the Hall Award, because all the other awards of the society are for people who are dead. That wasn't a good precedent. But anyway, that's what it is. So I really am delighted that you have gone outside the... I could do the lights from here, I think. Yeah, I've got a system set up. So I'm delighted you went outside the University of Massachusetts system. There are so many people that you could have brought in to give this talk. And so what Craig asked me to do, I think, was to talk about organisms and organismal biology and examples that I found interesting and still find interesting. And so I'm going to do that, and because I don't know anything except about the skeleton, I'm going to talk about the skeleton, and hence the Hence the title of the talk. Oops, systems. There are lots of colored slides, so I'm going to take the liberty of keeping the lights down, but not off. So Craig said choose a title that will attract people from the general public in, so I did. This is it. I don't know whether there's anybody from the general public here, but if there is, you might struggle a bit. Okay, so the skeleton, as most people know, it's either a woodcut that's hundreds of years old, or it's a Halloween sort of skeleton. It's an articulated set of bones. And that's the conception of many people of the skeleton. But when you start asking about skeletal systems, things get a little bit more interesting, because you can ask the question, how many skeletal systems are there? And I'm not going to ask you to raise your hands. The hint is that the number is somewhere between 1 and 4, which immediately indicates that, as in most studies and problems in organismal biology, the question is an easy one, how many skeletal systems are there? But the answer is actually quite hard, because it depends on what you mean by a skeletal system. So let's see what we mean. I'm going to first off start arguing that there are 3 skeletal systems. Can you hear me all right?
Quiet.
Not quite?
Quiet.
A little quiet. Okay. I'll try to be a little less quiet. So I'm going to argue that there are 3 skeletal systems: an axial skeleton, a craniofacial skeleton, and an appendicular skeleton. And I'll just quickly run through each of these. They're obviously based on position within vertebrates. So obviously we're talking about vertebrates. We're not talking about all the many invertebrate skeletons. So we have the axial skeleton, which consists of the vertebrae, and in many animals also consists of a tail, which can be interestingly divided and subdivided. And you can see the axial skeleton in a prepared fish. And so it's a vertebral skeleton that makes the backbone of all vertebrates. And it's axial because it runs down the middle axis. Then there is the craniofacial skeleton, which you can name in a variety of ways. It basically includes the skull. And I guess I should mention, for those who are not in the know, that anything that's blue is cartilage, anything that's red is bone, except for the background. I don't know why I still stick with the red background. So the craniofacial skeleton then consists of the skull, the jaws, there may be gills depending on the species. Here is a metamorphosing tadpole. So you have the craniofacial skeleton, you have the big brachial skeleton, you have the axial skeleton that you can see. So we have the craniofacial skeleton as a system which is separate from the axial skeleton, and it's separate geographically because it's in a different part of the animal, and it's separate for various other reasons. And then we have the appendicular skeleton, which of course in mice and humans consists of the forearms and the legs, and in fish consists of the pectoral girdle and the pectoral fins and the pelvic fins. And so these are the paired skeletons that you find in all vertebrates, in all fish, except for those that have lost them, which is something that we might say a little bit about. Okay, so this skeletal system, whether it's axial, head, or appendicular, is based in 2 tissues. It's based in cartilage, it's based in bone. This happens to be a fractured membrane bone from the side of a chicken, which has been repairing for several weeks. And even though it's a membrane bone, it's a bone which is not laid down with a cartilage model, it forms cartilage in the regenerative process. And so these 2 tissues, cartilage and bone, are very intimately related, and have been very intimately related for probably 500 million years. Okay, so those of you who came in earlier, this was a slide that was on when you came in. So this is the first answer to the question. We have 3 skeletal systems: axial, craniofacial, appendicular, composed of cartilage and bone. They're all inside the body, and so they constitute the endoskeleton. Is that okay now for noise? And so you can think of this as one skeletal system, the endoskeleton, based in cartilage and bone. Or you can think of it as 3 skeletal systems, depending on the position and the nature of the material that makes up those skeletal elements. But there's another skeletal system. It's reduced in most vertebrates. In humans, it consists of the collarbones, the teeth, and much of the craniofacial skeleton, much of the skull. It's much more extensive in many fossil vertebrates as a body armor. So that immediately implies that this is a skeleton which is on the outside. It's an armor, it's not an endoskeleton. And we know a lot about it from this fascinating guy, Bashford Dean, who lived from 1867 to 1928, who was simultaneously, in the 1920s, professor of zoology at Columbia University, where he worked on fish, curator of fishes at the American Museum of Natural History, and the first curator of the department that he created on arms and armor, the department of the Metropolitan Museum. And so here was somebody who worked on the exoskeleton, the skeleton that makes up the outside of many more basal vertebrates, and who had this fascination with armor. He had an enormous armor collection. This is Bashford Dean in one of his suits of armor. Here he is at the time that they established a special celebration of his work at the museum. So a really interesting fellow to whom we owe much of our knowledge about what we now call the exoskeleton. So here's an early Devonian jawless fish, a placoderm, and you can see that the upper part of the body is covered by this dermal armor, it's covered by this shield. And this is the state fossil of Western Australia, Macnamarapsis, which again consists of large amounts of dermal armor overlying this very extensively ossified skull. So the exoskeleton is not based in cartilage and bone. It's based on what we now think of as tooth-like elements, which are called odontodes. The terminology is not critical for what we're going to say. And it's based on three tissues, one of which is dentine, which tooth is now based on, one of which is enamel, which tooth is now based on, and the third is a basal bone, a bone that anchors the tooth or the odontode into the jaw, or in the case of the odontodes, into the skin. So you have this external skeleton, which is based on these tooth-like elements that were either individual elements within the skin, capped with dentine and enamel and underlain by bone, or they all fuse together to make an external armor. So if you look at the histology of this external armor, when it's all coalesced together as a single piece, you see these four different layers. The basal lamellar bone, and that's what's fascinating to me when I first learned about this, is that the histology on this, if you look at the histology, it looks exactly like the histology of modern bone, dentine, and enamel. This is obviously a diagrammatic representation. But you have basal lamellar bone, and you have spongy bone full of blood vessels and cavities. You have dentine, which you can identify because of the dentine tubules, and you have enamel on the surface. So it's very different from the endoskeeleton, which is based on cartilage, which is then replaced by bone. This is based on dentine and enamel, which is supported by bone. It's a very different system. And this is just to illustrate that what we have, and many vertebrates have as a remnant of that exoskeleton, are the teeth. So here is a very simple monocuspid tooth of a Mexican axolotl with the layer of dentine. The enamel doesn't stain here, just a few cells at the base. Here is the human tooth, and you can see the enamel, dentine, pulp, the basal bone of attachment, into which the tooth is supported. So we have these few remnants of the exoskeleton. They're inside the body now. They're teeth, and the collarbone, which we'll talk about a little bit later on, and elements of the skull. Okay, so we have, this is just to summarize that, this is the end of the first part. The exoskeleton, which is based on dentine, enamel, and bone, sitting on this basal bone, which you can regard in terms of thinking of the teeth. You can think of the bone of attachment as the alveolar bone that attaches the tooth into the jaw, and you can think of the basal bone as the jawbone, maxilla or mandible. And then we have the endoskeleton, which primitively arose as cartilage. That cartilage then became surrounded by bone, it calcified, became surrounded by bone, and then eventually bone invaded the cartilage and replaced it. So that's a developmental series that we see in vertebrate embryos, and it's the evolutionary history that we see through the fossil record. Cartilage, mineralized, surrounded by bone, invaded by bone, replaced by bone. Okay, so what are the various ways in which you can study the skeleton? I just thought I'd mention a number. You can look at growth and morphogenesis. I always argue that developmental biologist, which is what I started out on, until I was invited to think about my work in an evolutionary context, which I spent 6 months doing, and that was what produced that 1975 paper, because I'd just been doing development up until then. The three processes that developmental biologists are concerned with are growth, morphogenesis, and differentiation. Those are the three fundamental processes. How do things get bigger? How do things change shape? How do things become specialized? How do they differentiate? And so there are lots of ways of studying growth and morphogenesis. Here's one, it just shows the effect of head binding in various native tribes, the way in which you can distort the growth and shape of the skull in ways that are culturally relevant. And so by the right sort of binding, you get the right sort of shape for the skull with the subsequent consequences on the brain that fits the cultural context. And there are clearly many, many other ways of studying growth and morphogenesis. This is one rather dramatically. You can look at mutations as an excellent way of getting at differentiation and development. And so here's an individual who, aside from, has two abnormalities. One is the hair, which is probably not important. The other is he's missing his clavicles. And so he can approximate his shoulder blades together because the clavicles are missing. So here's an x-ray of a normal individual. Here's an x-ray of this individual with just these little tips of the clavicles present. Everything else in the skeleton is normal. So this is knockout of a single gene, RONX2. And it illustrates the modularity of the skeleton. That the rest of the pectoral girdle, in which the collarbone sits, is perfectly normal. The craniofacial skeleton is normal. All the rest of the skeletal systems are normal. This one bone has been affected. And it turns out that that one bone, the clavicle, is the one membrane bone. It's the one exoskeletal bone that moved inside and is associated with the pectoral girdle. So the rest of the shoulder girdle is in fact endoskeletal, but the clavicle is exoskeletal. It's coming from the outside. And in kangaroos and a few other groups, there's an equivalent bone in the pelvic girdle as well. I put this, do you do a triathlete? Are you a triathlon or are you just a runner? You're obviously slim, but you're just a runner.
I don't bike.
You don't bike? Okay, that's probably very sensible. This guy biked and swam. This is what happens if you come up your bike at very high speed. I didn't put the picture in that shows this 10 pins and a large plate that was needed to stabilize this clavicle. But it's easily broken, but usually not a problem. In this case, it was a fairly substantial problem. So this tells us something then about the origins of the clavicle, and it tells us something about this concept of modularity. That within a skeletal system, within the pectoral girdle, there are modular elements. And in this case, one modular element is this bone that came from the exoskeleton. It turns out that if you look within a single bone, this is the dentary, the single bone that makes up the lower jaw of mammals. If you look within the dentary, it's a single bone, but it's a very complicated single bone, which has a whole variety of different morphological areas. This is from a mouse, and so it has the mouse, of course, have reduced dentition. They have incisors and molars and a big gap in between. So you have the incisor bone, which has this long root, takes up much of the body of the mandible. You have the molar teeth, and then you have these three processes at the back of the mandible, which are actually developed in a way which is very different from the rest of the body. But I put this slide up just to show that if you knock out individual genes in mice, you affect particular parts of this single bone. So if you knock out MSX1, for example, you affect what are called the alveolar units, the units associated with the teeth. If you knock out goosecoid, you affect two of these processes. If you knock out TGF-beta2, you affect all three processes. And so you have different genes acting differentially on different parts of the skeleton, because each part of this bone is made from a separate population of cells. So you have modularity within this single bone, I think probably most dramatically shown by the mandible, which is why Bill Axley and I in 1991 used the mandible as the bone for a model for how you could analyze development and morphogenesis through evolution and through developmental time. And so the front part of the mandible is very much influenced by the teeth, whereas the back part of the mandible is very much influenced by muscle action, because the major muscles of the lower jaw, as you can see in this not mouse, but something else, you can see attached to these three processes. So the posterior part of the mandible is very much influenced by muscle action. So the action of TGF-beta might in fact be directly on these cells of the bone, or it might be in fact influencing the muscles, so that you have an indirect effect on the skeleton. And in fact, if you knock out MIF5 and MyoD, if you knock out the two genes that control muscle development in mice, you have substantial effects on this part of the dentary, some from the knockout of the effect on the muscle, but some from more direct effect on the elements themselves. So this concept of modularity is becoming an important one in evo-devo, the notion that you can take a part of an organism and look at it as component parts, which are differentially acted on by genes and which are differentially acted on epigenetically, in the sense of epigenetics in the broad term of environmental influences controlling development. In this case, it's broadly muscle action at the back, action of teeth at the front. Okay, so when we think about skeletal development in ourselves, for example, when we teach human anatomy, we think of most of the skeleton developing fairly synchronously. So if you look at this human embryo, you can see that the craniofacial and the axillary and the appendicular skeleton are all at relatively similar stages of development. They're all made of bone now, there are some small elements of cartilage left, but development is quite synchronous. That's not true in organisms which have a larval stage in their life cycle. And so you can uncouple these skeletal systems, and that's most clearly seen, I think, in amphibians. So in animals that have a larval skeleton, let's say a frog which has a tadpole where the skeleton is completely cartilaginous, and the adult frog where the skeleton is almost completely bone, you have two quite separate skeletons at two different stages in the life cycle. You have the larval skeleton, which is cartilaginous, and the adult skeleton, which is bone. And here you can, and I showed you this slide before, you can see the cartilaginous elements of the skull, you can see the branchial cartilages of the gills, they will disappear during metamorphosis. You can see the very beginning of forelimb development, the cartilages of the humerus and the digits, and the very beginnings of axial development. And so one question that's interested biologists for some time, is where in the tadpole do the cells come from that make the adult skeleton? So if this is going to disappear, and if the cartilages of the tadpole are going to be either replaced or turned into cells of the adult, then where do the cells of the adult skeleton come from during development? And I'm just going to, I think I can put, well, I guess I do need to show you this. This just shows those same two images at a later stage of development of the adult skeleton beginning to appear with the ossification of the vertebral column. This just shows that if you look at the limbs, it's fairly obvious where the limbs are going to come from in the frog. They come from a specific area of mesoderm in the same part of the body as the limbs of any other tetrapod come from. The head skeleton is rather different. So this is, for those of you who know about this stuff, this is mesodermal in origin. Most of the endoskeleton is mesodermal. But the head skeleton, this is the head of an embryo. You can see the eye, the mouth would be here, going around to the midbrain, running down to the body. The head skeleton comes from these cells that migrate out from the developing brain, from the neural crest cells. So the head skeleton has an entirely different origin from the appendicular and the axial skeleton. It comes from neural crest cells. The neural crest cells are the basis of the exoskeleton. And so these cells migrate down into, say, the lower jaw. And so a question that one wants to know in tadpoles is the cells that are going to make the adult skeleton, that say migrate down into the jaw and are going to make the bones of the adult, where are they during metamorphosis? And the short answer is we don't know. People have started to look. And I've addressed this just by raising a number of possibilities, because depending on the skeletal element in the adult that you look at, all of these turn out to be true. So it's not just a simple replace the cartilaginous skeleton with bone. You can have the tadpole cartilage becoming an adult cartilage. And that's what that, I won't give you examples of where that happens, or I'll run out of time. Well, I mean, I hate Knuckles cartilage. Is an example. The tadpole cartilage can be replaced by or turned into an adult bone. That's a second possibility. The tadpole cartilage might disappear, like the gill cartilages, and not be replaced by anything, because the adult doesn't have gills. The tadpole cartilage disappears, and a new cartilage or bone forms in its place from a different group of cells than those that formed the tadpole cartilage. Part of the tadpole cartilage turns into the adult cartilage or bone. And that typically happens when cells in the perichondrium. The perichondrium is the layer of stem cells and progenitor cells that surrounds the cartilage. Some of those cells can be sort of set aside in the larva to make the skeleton of the adult. So that's this notion. So those cells are set aside in the perichondrium, in the layer around the cartilage. There are also, we think, although we haven't, nobody's identified these yet, we think that there are other set-aside cells in the tadpole that are these neural crest cells that migrated down. They're now sitting in the tadpole waiting to become an adult cartilage or an adult bone. And this all applies just to the craniofacial skeleton. The axial and the appendicular skeleton are formed by mesoderm in the standard way. So one of the reasons why organisms are important to me, and still important to me, is that we've answered virtually none of these questions for any amphibian. We know a little bit about some of the adult bony elements from work of Jim Hankin and his group at Harvard. Is it all right to talk about Harvard here in Amherst? At Harvard. But it's been very tricky to get markers, to get transgenic lines that would take you far enough into the adult stage to look at what happens in late stages. Okay, so what if there's no tadpole stage? What if you're a frog that doesn't have a tadpole? And there are hundreds of such frogs. They're called direct developers. And so they hatch as small frogs and develop into larger frogs. And the best studied example is the Costa Rican coqui, the Luthrodactylus coqui. So these are animals in which the larval stage has been lost from the life cycle, which always sounds to me like a very hard thing to do evolutionarily, to drop a whole stage out of the life cycle. But in frogs, it's probably happened 7 or 8 times independently and affects 700 or 800 species. And so here is a scanning electron micrograph of a coqui embryo. You can see, if you're used to looking at scanning electron micrographs, you can see the head, you can see the eyes, you can see the body, you can see the large amount of yolk on which this is sitting, and you can see at this very, very early stage in development, when the neural tube is just beginning to close over posteriorly, here's the forelimb bud and the hind limb bud. They're already there. So you have this tremendously accelerated development in the direct developing frog of the forelimb buds and the hind limb buds, because remember the metamorphosing species are very different. And these two preparations came from Jim Hankin, again at that other institution. And this is the collagen types 2, which stands for skeletal elements. And so you can see how the axial skeleton is developing very early. The limb buds are already present and in fact laying down cartilage. So here's this animal that still has this large yolk sac, it's still in its very early stages of embryonic development, but it's already laying down a humerus, radius, and ulna, and digits. So you can uncouple skeletal systems from one another, cartilage from bone, and you can uncouple skeletal systems in time, and that's really nicely illustrated by coqui. And I put this in just for completeness, just to say one slide's worth about limb loss and transformation. Because clearly many tetrapods, although they're called tetrapods, that is, they have 4 pods, they have 4 legs, they are in fact limbless. And so snakes, which have no legs, are tetrapods because their ancestors were tetrapods. And so you can in fact lose a character that's diagnostic for the group you're in, but still be part of the group. And one very nice example that's beginning to be looked at are whales, where the forelimbs become transformed into flippers and the hind limbs completely disappear in the adult. But the hind limb buds, the forelimb bud is present in these 2 embryos. You can see it's developing out into what will be the flipper. You can actually see some of the digit primordia there. But there is a hind limb bud in this early stage embryo. So losing the hind limb in the adult doesn't mean that you lose the ability to make a hind limb bud. That bud begins to develop, and this is the case in most snakes and in legless lizards and in most limbless tetrapods, that the hind limb, the buds begin to develop and then they regress. Cell death ensues, and the cell death ensues because there's a population of cells that should migrate down from the mesoderm into the limb bud, and that population doesn't arrive, it gets killed off, and that population produces FGF8, which is required to maintain the limb bud. So in the absence of FGF8, you lose the ability to maintain the limb. And that mechanism seems to be true for most limbless vertebrates. Every now and then, I'll tell you about the next slide. There are some whales, of course, that have a rudimentary element of a hind limb piece of skeleton. The limb buds continue to develop, and you may get something which is variously called a femur, or it's been given other sorts of names because it's been somewhat modified and seems to be associated with muscles involved in reproduction, so it's not quite clear what its origin is. But what is clear is that every now and then, and in sperm whales it's about 1 in every 5,000, the limb buds continue to develop and can in fact produce something which is quite a respectable hind limb. So here is a dolphin, here are the flippers, and here is this quite prominent hind limb which is developed. And the possibility of that development is because the limb bud was present, something has presumably overridden that cell death that would kill off the limb bud, and the limb bud has not only continued to develop, but has in fact been able to produce an articulated set of skeletal elements. Okay, now the first part of the title was this fish that climb waterfalls. And so let me tell you a little bit about fish that climb waterfalls. And I know there's somebody in there who's an expert on this group. And in fact, when I looked at the website for the OEB program, I realized that a number of things I'm talking about which I know nothing more than what I'm saying here, there are experts in the audience. So bear with me on that. If you're a generalist, you suffer from being a generalist. Okay, so what's this amazing transformation? This is the first photograph my wife ever took when we left Australia. It's, I'm going to talk about fish that are in Hawaii. Those of you who know the Pacific Islands, this is not Hawaii, it's Tahiti, but one Pacific Island is a bit like another Pacific Island. So this was the first photograph we took on the way to our 2-year appointment at Dalhousie in 1968, coming into Tahiti, which is filled with waterfalls of all sorts of sizes and shapes. There is a group of Hawaiian fish, locally known as the oopu. And this is one of the more prominent species, Latipes concolor, which has a pair of pectoral fins, so it has the equivalent of the forearms, but it has no pelvic fins. It has no equivalent of the legs. But what it has is a midline sucker and very fleshy lips, and no pelvic girdle, no pelvic fins. And so here is Oopu nakea, which is the largest and most abundant of these species of gobid fishes. And you can see the prominent pectoral fins, and you can see the single midline sucker. And so there's a sucker present in the midline, there are pectoral fins, but there are no pelvic fins. The life cycle of the oopu is very interesting. Spawning peak is in February, in nests which are close to stream banks, in fresh water, often at the top of quite high waterfalls. The larvae hatch upstream, and they're flushed down and into the ocean, because these are fish that need to continue their development in the ocean. So that at the embryonic stage, they're flushed down out into the ocean, they continue developing in the ocean. After developing at sea, they return and they climb back up these waterfalls to get back to the rivers where they were spawned. So these are the fish that climb the waterfalls, and you can see one here with its sucker. And I've just shown one here with its sucker. So basically what they do is they alternatively use the mouth as a sucker and the sucker as a sucker to sort of leapfrog up the waterfall. Often several hundred meters, I mean really quite dramatic. And so I've said that the sucker is present, but the pelvic fins are not. And that's because the sucker is derived from what would have been the pelvic fins. Pelvic fins have moved to the midline and fused and become a sucker. I'll just illustrate that with a couple of species that we've done a little bit of work on in Halifax, just to show that the form of the sucker can vary enormously. This is a large, very mucus-filled sucker. Here is one that's very much like the one we see in the Hawaiian forms, circular, very good for sucking onto a surface. You see something similar in some of the fishes in the Tibetan and other fast-flowing rivers. If you look at the structure of the sucker in side view, here's the craniofacial skeleton that we saw before, here's the actual skeleton. Here's the sucker in side view, which doesn't tell you very much. But if you look at the sucker ventrally, you see that what it's made out of is fin brays from the pelvic fin, and a central element which is derived from the pelvic girdle. So here's an appendicular skeletal element which has moved to the midline, fused, and been modified into serving a different function, that of a sucker. So it's an amazing transformation. There's been very little developmental work. A lot of these species are protected now. There's been very little developmental work done on these. It's a fascinating system to ask questions about homology, because the argument would be that if the sucker never coexists with the pelvic girdle, and we know that the pelvic girdle is in fact modified and fin is modified into the sucker, then the sucker and the pelvic girdle are homologous structures. Even though they're serving different functions, they're homologous in the sense of characters within the organism. So the basis then of this comparison between pelvic fins and midline sucker is this essential similarity, this sameness of characters that you see between apples and apples, and that you see between wings of bats and wings of birds, although I realize neither of these images show the wings very well. And it's this central concept of homology. And I think arguably evo-devo has had a fairly substantial role in bringing forth homology as a concept which goes way back and was really modernized by Richard Owen in the 1840s as bringing homology back into a way in which it could be analyzed mechanistically in development and evolution. So similarity then was rational. These are two images of Richard Owen, who was the first director of the British Museum of Natural History. He is normally depicted in old age as a sort of gnome-like figure. Everybody hated Richard Owen. Darwin hated only one person, and that was Richard Owen. But as a young man, he was actually quite, I think, attractive. Okay, so the staunch anti-Darwinian, anti-evolutionist Richard Owen, who left us this incredibly important and foundational legacy. It's involved whenever we make comparisons between two or more biological objects. And in my view, it's the greatest legacy of any non-evolutionist to modern biology. And it's this concept of homology. So the criteria to reveal homology weren't changed by Owen. They were developed back in the 18th century by a French anatomist, Étienne Geoffroy Saint-Hilaire. And there are three criteria that were and are still used to determine the homology